Good morning.
Good morning.
Good morning. All right. Yeah. Yeah. Let's chat.
Good morning.
Morning. Good to hear from you.
Yes.
Yeah.
Oh, it's echoing.
I think you probably got some other sound source on. There we go. All right.
Good morning.
Morning. Good to hear from you.
Yeah. Do you know when I get my access code?
They came late last night. I'm going to give them to you today.
Oh, dope. All right.
Yeah.
I thought that Jim said that we're not supposed to be using Connect.
I was able to get the access code, so we're good. So you'll be able to get to all your
Okay,
assignments
because
and complete them.
so none of the stuff is going to be marked late because we couldn't get on there.
No late penalty. No, because again, the problem with Connect was on their end. So, all right. So let's do a resend. Resend. Okay. Reset.
Hey, Mr. Richard, this is
Yeah.
Melissa.
Yes.
Where do you go to get help with the lab?
Well, you can come to tutoring, which is Friday from 5 to 7.
I have work on Fridays. It's impossible for me to come into tutoring.
You work past 5?
Yes, I work 7 to 7.
7 to 7. Okay.
Yes.
Then what we would have to do is have special session, okay, that works with you. So you'd have to tell me what works, and then we could schedule that, and I could set that up. Okay. All right. Let's see.
Okay. And this is Melissa
Good morning,
again.
everybody.
Morning.
This is Melissa again. Do we have to purchase the book?
Well, use the code today
Okay.
and see if the ebook shows up. It should show up.
Okay.
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 Yeah. Today. Attached. All right. So, okay. So lecture notes are in chat. I'm sending it to you also right now. Da, da, da. There you go. Here come the lecture notes in your email. All right. Okay. Access codes, access codes. Okay. So each one of you are going to get a unique access code. I'm going to send that access code to each one of you separately. Okay. All right. So. We are recording, so we're good. And we'll start with chapter 11. Let's do a share. Yes. Come on. All right. Share. There we go. All right. Now we're ready. Okay. Okay. Yeah. There we go. All right. All right. So today we're going to talk about the interactions between you and your guests. Believe it or not, your body is serving as a hotel for all kinds of organisms.
I have one more question.
Come again?
How do we see what our current overall grade is in the class?
I haven't input that stuff yet because I haven't had your access codes yet. Okay. So I'm going to get all that in today. So that'll come in. You'll be able to see everything. Okay. So we're going to talk about how you interact with the environment around you in terms of serving as a host, right? You've got stuff growing on you, in you, and around you all the time. All right. And you're in a state of equilibrium with those organisms because there are organisms out there both that cause disease and those that don't. And the ones that don't cause disease, in many cases, protect you from the ones that do. And they're able to do that by crowding out room for the pathogens to try and grow on you and in you. Okay. And they do that by stealing space and stealing nutrients and creating an environment that favors them over the pathogen. If you're healthy, you coexist with your microbes and you don't get sick much. But occasionally the balance will tip, and those disease-causing pathogens that are always out there are going to come in and try and change your state, right? Try and produce disease. So the Human Microbiome Project was just an effort to try and figure out what's living on you, in you, and around you, okay? And what they did was they just looked at the DNA from the things that are living on you, in you, and around you and compared that to the DNA in your body. And what they found out was that there were 22,000 protein encoding genes in the DNA that they checked, while the things that are living on you, in you, and around you had about 8 million. Okay, so that's a huge difference. And the point is that you've got a lot more stuff growing on and in and around your body than we previously thought, even in places that we used to think were sterile, meaning there's no bacteria in there at all. All right. Healthy people have dangerous pathogens on them and in them in low numbers, but these other microbes keep them in check. Okay. So we have all kinds of environments on our body, right? We've got moist, dark, warm environments. We have dry, salty, warm environments. We have acidic environments. We have basic environments. Okay. And most areas of the body that you can think of are in contact with the outside world and have microbes in them. And so these would be things like your digestive system and your respiratory system and your urinary and your reproductive system, as well as things like your orbital cavities where your eyeballs are housed, okay, your ear canals, all right, and even maybe your middle ear because it's connected to your throat. And those are places that we would expect to see stuff because there's a direct doorway to the outside world. It's always open for all of those systems, right? Your digestive system is open at both ends, at your mouth and at your anus. Your respiratory system is open at your mouth and your nasal cavity. And your eyes are open to the outside world. Obviously, if they're open, even if they're closed, really, they're accessible to the outside world. And of course, your urethra is open to the outside world, and that allows passage up the urethra and say into the bladder, and in some cases, even up to the kidneys. Okay. And those are all things that we expected. Okay. Most of the microbes that come in contact with our body get wiped out by our immune system before they get a chance to get into the blood and cause trouble. But some have developed a relationship with us in which they can survive and not hurt us. Okay, so they can hang out and everything's okay because they don't do anything to upset the normal function of our body systems. Now, sometimes what happens is that the mechanism used by our useful microbiota, meaning the good bacteria or the good microbes, is just to take up room that normally pathogens would try to use. So what they do is they steal all the space that's available. There's no room left for the pathogens to attach and cause trouble. Okay. And that means that the normal bacteria, the good guys, have to have places to hook up and have to be able to stay happy and healthy in those places. All right. Factors that weaken our defenses and increase the likelihood of an infection are old age and extreme youth. Now, you might wonder, well, why would that be? Well, that's a good question. Okay. And the answer turns out to be a weak immune system. Okay. Genetic defects in immunity and acquired defects also mean that we have a weak immune system. So that really qualifies for both of these, right? Surgery and organ transplants. And the reason for that is that the body is in a weakened state. Body is trying to recover from trauma. Right? They cut you open, they work on you, they sew you up, and the body has to heal. Okay. And in the process of doing that, stuff that normally helps out your immune system might be used for things like wound healing, namely making protein. And the result then is you might end up with an infection that you normally wouldn't pick up. And then things like cancer and liver malfunction and diabetes also weaken your immune system. Okay. Now you might think, well, why is that? Well, your liver makes proteins in the blood that defend against attack called complement. And if your liver is hurting, then you don't have as many of those proteins in your system and you're more likely to get an infection. Okay. Diabetes produces damage to internal organs that has to be repaired. And it also is going to mess up protein metabolism, which means it's harder for you to make white blood cells and harder for you to make antibodies. And finally, in cancer, what happens is that the cancer cells take over the bone marrow, and the result then is that you can't make normal white blood cells to fight the infection, right? And the same happens if you're on chemo or if you're on immunosuppressants for the treatment of something like cancer or autoimmune disease, right? Is that your immune system is weakened, okay? Stress is going to weaken your immune response because it increases levels of cortisol, which is a hormone that's made by your adrenal cortex that has as one of its effects suppression of inflammation. Increased cortisol means reduced inflammation, right? And if that happens, remember that inflammation is actually a good thing. It's bad in that it hurts, but it's good in that it is bringing the white blood cells to the area where the trouble is so that they can do their work. And if we're not doing that as effectively, you're more likely to get sicker quicker. And then pregnancy, okay? And the reason for that is that you're causing a lot of resources to be diverted to building the baby as opposed to protecting you against illness, right? So those are all things that are ultimately going to suppress the immune system either directly or as a secondary effect, right? And with less immune response, you're more likely to get disease, okay? Until recently, the uterus and its contents were thought to be sterile, but analysis of newborn stool before their first meal showed that there were bacteria in there, showing that their intestines are colonized while they're still inside their mother. And you might wonder, well, my goodness, how is that possible? Well, it probably came in through the placenta. There were probably some types of bacteria that were able to get past the filtration apparatus of the placenta and get into baby's blood and then colonize baby, okay? Comprehensive exposure occurs right after birth and during birth. As you pass through the vagina, you get colonized by lactobacillus and prevotella and other bacteria that are in the birth canal. The lactobacillus lets the baby have enzymes that help it break down protein in milk, and other bacteria are going to help the baby not suffer from skin problems and other conditions. Human milk has about 600 species of bacteria that are in it. You might think, my goodness, I wouldn't want to drink that. I might get sick. But all those bacteria turn out to be either neutral or beneficial. Okay. So they're good for you. They're the good guys. Right. This same project showed a lot of variation in our normal microbiota, the good bacteria that live on our body. So everybody among us, right, has a normal collection of bacteria that live on them, in them, and around them. But from person to person, which bacteria those are can be different. And even in a single person, you can have different bacteria on one region of the body versus another region. Okay. So more important than what's living on you is what it is they make that keeps you healthy. All right. So they're talking there about the proteins and the enzymes that they generate that keep the pathogens from doing their work. So there's at least 100 types of fungi in the intestine, 1 billion viruses in the feces, right? So you're far from in a sterile condition. We're going to talk about what causes a microbe to be able to get you sick, okay? And that's called its virulence, all right? Its virulence is its ability to make disease. Okay. And those weapons that allow it to do that are called virulence factors. Okay. So think of those as disease making weapons. Disease making weapons. We're going to talk about opportunism, and then we'll talk about how a microbe gets you sick if it's a pathogen, and talk about how they get in and out. Those are called portals of entry and exit. Okay. Then we'll talk about infectious dose and talk about how that produces an infection, and talk about ways that they cause tissue damage. How do they wreck what's good inside you? Okay. We'll talk about toxins, talk about the virulence factors. We'll diagram the stages of disease in a human, and we'll talk about different kinds of reservoirs. What is a reservoir? Okay. A reservoir is a place where living microbes are found. Okay. And then we'll talk about different modes of transmission, right? Mode of transmission is how microbes travel. Okay. And then we'll talk about nosocomial infections and tell you what kind are most common. And then we'll finally get to Koch's postulates. Okay. So a pathogen is just any microbe that makes disease, okay, which is any change in your health that is going to produce tissue damage or symptoms that are uncomfortable or painful or damaging. Okay. The pathogenicity of the organism is the ability of that organism to make disease. Now, some organisms have high pathogenicity and some don't have any, okay, which is going to talk basically or speak to the weapons that they have. So some organisms that are extremely pathogenic can make you sick with a very low dose. And those that aren't pathogenic can't make you sick at any dose. Okay. A true pathogen makes disease in somebody who's healthy. All right. Which can vary from severe to fatal. All right. And then an opportunistic pathogen is one that makes you sick if you have a weakened immune response. Okay. So I have to differentiate between those two. What you're looking at here is just an example of different outcomes, right? You've got on the diagram here the name of the microbe, right? The host, and then finally what happens, all right? What's the result of all this? Okay, so hold on here. I just want to. Showing it to me. What you're seeing, okay. Sorry, go back. What you're seeing in this diagram is just an example of the ability of the microbe to produce disease being dependent on getting into the body through the correct door and then being able to get by the immune system and then get into the tissue and start wrecking you. Okay. And that only happens in the third scenario from the top, right? Where you got the two off buttons gone to red. Okay. And the result then is, sorry, fourth scenario where you've got the first button is green and the second button is red. Okay. And that produces disease. And what they're saying down there in the bottom is that the microbe got in through the right door and the microbe had never been seen by the immune system before. And the result is that you get sick. Okay. In every other situation, what happens is that you avoid disease, either because the immune system picks it off or the microbe either doesn't come in at all or doesn't come in through the right doorway. Okay. So an opportunistic pathogen is one that causes disease when our host defenses are weakened, right? Our immune system's not doing its job right. And it's not considered disease causing to somebody who's normal and healthy. And so these organisms don't have powerful virulence factors. And examples include things like Pseudomonas and Candida that are living on us and around us all the time, but normally don't make us sick because the immune system keeps it in check. The virulence is just the severity of the disease that the microbe produces. And what we do is we rank its degree of pathogenicity, right, as a measure of how easy it is for that microbe to make you sick. Virulence of a microbe is going to be determined by its ability to colonize you, to damage you, right, and then to avoid being destroyed by the immune system. And so a virulence factor is any characteristic that lets it do that, okay, which be modifications, for instance, that let it hook on and colonize you, or modifications that, if it's free floating, say in the blood or in the tissue fluid, cause damage to the tissues around it, such as endotoxins and exotoxins, okay? So you have to go in through the correct portal of entry in order to make disease, right? And that's just a doorway into the body, which could be what? An opening in the body, such as your nose or your mouth or your ears, right? Or even your anus or your urethra, okay? An exogenous microbe comes from outside the body or another person or another animal and gets on you and in you and could cause disease, while an endogenous one always is living there, but manages now to get into an area of the body where it can cause trouble where before it couldn't, okay? An example would be like the staph that lives on the surface of your skin. Normally the staph doesn't cause any trouble. But what happens sometimes when you get a cut or an injury is that the staph can get under the skin and even in some cases into the blood and can cause sepsis and tissue damage, okay? But as long as you don't have an injury, then it's locked out because the normal properties of the skin keep it in check, including the good staph that lived there, okay? So the portals of entry, we've kind of talked about, right? Wounds or systems that are normally open to the outside, right? So if you have a puncture or cut, then they can get in through that. Obviously, you've made a new opening. So they can get under the skin and even in some cases into the tissue fluid and in the blood, okay? But if they go through the wrong doorway into the body, say something that normally causes problems in the digestive system gets into the respiratory system, then it's not going to cause you trouble, okay? Because it got into the wrong place and it can't produce the damage it normally does, all right? Something that might cause a respiratory infection might not cause trouble if it got in through a wound, okay? Because that's not normally where it likes to be, where it likes to thrive. But sometimes a pathogen can get in more than one way, right? The bacteria that causes tuberculosis can get in through the respiratory and the digestive tract. And streptococcus and staph can get in through the skin, the urogenital, or the respiratory tract, okay? So there's different numbers of doorways that some of these bacteria can use to cause trouble. The size of the dose is critical, right? So we call this the infectious dose or the ID, right? That's the minimum number of bacteria that we need to make you sick. Microorganisms with a smaller infectious dose have greater virulence, right? Because they have more virulence factors and more potent virulence factors. So the ID for rickettsia is one cell. You know, you get one cell on you, you're going to end up with those GI conditions, okay? But for cholera, it's millions of cells, okay? So you're much safer being exposed to the bacteria that cause cholera than you are to the pathogens that produce rickettsia, right? That's why we take different precautions with different pathogens. Next step after you get in is you have to latch on, right? If you can't latch on and build a home, build a campsite, then you're not going to be able to grow and thrive, right? So this is how the microbe grabs onto you and hangs out, right? It depends on binding between surface molecules on the pathogen and the host, right? So they have to have the right grippers and you have to have the right surface for a hookup to happen, or else it's just going to slide right through. It's going to be flushed out of the system. The pathogen is only going to colonize those tissues that it can hook onto. This is important for it to cause disease because the body has mechanisms to flush out stuff that's not latched on. Secretions, for instance, on your skin, like your sweat or your oils, can flush the surface of your skin. Mucus can flush your respiratory and your digestive system and your urogenital system, okay? So how do they hook up? Well, they can use Fimbriae, they can use surface proteins, and they can use slimes or capsules. Viruses latch onto particular receptors, such as the AIDS virus using CD4 on helper T cells. Parasitic worms use suckers, hooks, and barbs. So they latch on, okay? Bottom line, you have to latch on to cause trouble. When we have to survive our defenses, right? Phagocytes are white blood cells that run around our body eating up invaders through endocytosis and then destroying them using enzymes that are inside the lysosome, okay? That kills them, and then pieces of that are produced on the surface of the white blood cell and used to warn the rest of the immune system that you have an infection, okay? So if we can avoid this, then we can avoid getting wiped out before we make disease, right? So how do they avoid getting eaten? Well, there are virulence factors that let them avoid things like macrophages, such as leukocytins that kill phagocytes straight up, right? So they spray our white blood cells with these things and they die, right? And then a slime coating or a capsule makes it hard for the phagocyte to grip the pathogen. Okay, so it tries to eat it, but it slips away. Okay, so it either can't grab it or we kill it before it gets a chance to grab it. All right, and then some actually live inside the white blood cell and use it as a place to reproduce. So that's really sneaky, right? They're inside the white blood cell. Normally they'd be getting chewed up, but because of certain weapons that they make, the lysosomes can't come in and destroy the ingested microbe, right? He hangs out until he produces enough of himself to either rupture the white blood cell or leave from the inside out and cause trouble. And then how does it actually cause the disease, right? Once we've gotten in the right door, once we've knocked back the immune defenses, how do we make you sick? Okay, what is it that we have that causes disease? Well, we can use enzymes that tear up normal protein in our body. Okay, we can use toxins that produce a response that damages our cells and tissues. Okay, so a toxin is different from an enzyme in that it's not going to cause a chemical reaction. It could cause, say, a membrane to get leaky, or it could attract our own immune system and cause damage to healthy tissues by leaving, say, decoy molecules behind. Okay, and it can do damage indirectly by having the host defense respond in an inappropriate fashion. All right. So the last two kind of overlap. Okay. So some examples of enzymes that make damage are those that break down and tear up our own tissue, dissolve our defense barriers, and let the disease spread through other places. Examples include mucinase that tears up the coating on mucous membranes, and hyaluronidase that tears up material that's in the extracellular matrix that glues our tissue cells together in connective tissue. Okay, so normally in connective tissue, say in the skin, you've got cells that kind of live in this matrix of protein and water and wandering cells and cells that are latched in place. But when you produce this enzyme, you tear up that matrix and the bacteria can then penetrate maybe even all the way into the blood. Okay. Coagulase makes blood or plasma clot. Okay. And you might think, well, how does that work exactly? Well, when you cause coagulation, then you actually make it easier for the bacteria to get the iron out of the blood that they need to grow. Okay. So this is a virulence factor in which you can coagulate and lyse red blood cells. The iron that comes out gets used by the bacteria and they begin to grow and produce even more damage. And you can pick this up on, say, a blood agar plate, right? A blood agar plate is normally red, but light doesn't pass through it. It's kind of red and creamy looking, okay? And when you grow bacteria colonies on the blood agar plate that produce coagulase, what'll happen is that you'll get a zone around the colony that's transparent, and that's called hemolysis. Lysis of red blood cells indicates the presence of things like coagulase. Okay. Kinase can dissolve clots. All right. And so what happens if they express kinase is they can get through the temporary barrier that our body uses to keep us from bleeding to death, and from there get into the blood. Okay. And then toxins, right? Products of microbes and other pathogens that are poisonous to the host, right? Neurotoxins target the nervous system. Enterotoxins hit the intestines. Nephrotoxins hit the kidneys. And hemolysins tear up red blood cells and release the iron inside. An endotoxin is one that is physically attached to the pathogen, such as attached to the bacterial cell wall, right? Lipopolysaccharide is part of the outer membrane of gram-negative bacteria and causes effects on tissues and organs that damage them, such as fever and inflammation and bleeding and diarrhea. Blood infections by things like Salmonella and Shigella and E. coli can be very dangerous because of the presence of endotoxin, right? So you can see here some examples of exotoxins and endotoxins and what it is that can knock them out and what effect they have on our body's defenses, okay? And also whether or not they get released, okay? Next, we have to look at how we spread the disease effects, right? Microbes eventually settle in a target and damage the site because the host tissues get weakened as the pathogen grows. Why is that? Well, number one, the pathogen is stealing nutrients from the tissue, okay? So it makes it tougher for the tissue to do its work, right? They also block the production of blood vessels and lymphatic vessels and bile ducts, okay? And the result then is that you don't have any pipeline to move material through the body in those areas that are infected, okay? And then they can cause tissue death, right? The damage that the pathogens make causes the tissues to die and can lead to even further spread of infection, right? Viruses wreck cells by blowing them up, right? So many viral particles build up inside the infected cell that it literally explodes, okay? So all these are ways to make disease. And so this is just a table showing you some of these different methods, all right? How it is they get in, get on, grow, and then toxify and damage the host. And then how do they get out, if ever, okay? So what are some warning signals of disease? Well, in A&P, you learned about signs and symptoms, right? A sign is anything that you can put a number on in a patient that indicates disease, such as what is your temperature, or what is your current weight, or what is your heart rate, or what is your blood pressure, okay? A symptom is a description of either the appearance or the feeling of the patient, right? So the patient gives you these details, right? And you can collect some of these details yourself. And a syndrome is just a collection of symptoms that indicates the presence of a particular disease, right? So let's take inflammation, right? Which is designed to protect us against infection. Symptoms are going to be fever, pain, soreness, swelling. The signs are going to be edema, granulomas and abscesses, all right? Lymphadenitis, and all of these indicate infection. So how is it that these are beneficial for us? Well, if we take the example of, let's say, fever, right? What's the objective with fever? To raise the temperature so much that the pathogen has trouble reproducing while the white blood cells are capable of reproducing, all right? So this tells you that something's trying to set up inside the body and your body's trying to fight it. The pain comes from the pressure on the nerve endings that produce pain. And that's the result of the swelling in the area, as well as the release of certain pain producing chemicals. Same for soreness. And then the swelling is just an indication of the collection of fluid in the area. And you'll also see redness, right, as blood comes in. The edema is the literal accumulation of the fluid in the area that's infected. Granulomas are inflammatory cells and microbes doing battle inside the tissue. So it'll often look sort of reddish and soft and spongy. And then lymphadenitis is just the swollen lymph nodes, and they get that way because they're collecting dead tissue cells and dead pathogen from the tissue fluid, as well as living pathogen, right? And as they bring all that stuff into the lymph node will swell up and become painful. In the blood, we see leukocytosis, which means that your white blood cell count goes up. The reason is you're trying to fight the infection. Leukopenia, which is a drop in white blood cell levels, which means that during the disease process, the pathogen has produced something that has caused the white blood cells to be less effective at getting into the bloodstream and defending you, okay? And then septicemia, which are microorganisms that are literally growing in the blood. This is very dangerous because this can lead to multiple organ failure and death. Bacteremia or viremia are bacteria or viruses in the blood, but that aren't actively multiplying, okay? Some infections don't get noticed because there aren't symptoms right away, or the symptoms aren't easily detected, right? The microbe's in the host, but the host feels fine and doesn't go to the doctor. And these are called asymptomatic or subclinical infections, okay? So these people can still spread the disease, but they aren't capable of suffering from the effects of the disease at that point. Okay. So let's take a break here. All right. And we'll come back in 15 minutes and get the second half of this. Okay. Okay, we are back. Okay. The last part of this is, how does the microbe escape? Okay. How do they get out of the body? Some of them never do. Okay. But most of them eventually leave. They can be shed through secretion, excretion, discharge, or sloughed tissue. Remember that your skin is regenerating every day, and the cells get shed off into the environment. And along with them are the pathogens that are living on those pieces of skin. An increased number of microbes in these materials makes it more likely that the pathogen is going to get to other hosts, right? So as you evacuate what made you sick, then it's available now for other organisms. The portal of exit is usually the same as the portal of entry, but some pathogens get out in different routes, okay? For instance, if you ingest a pathogen, right, it may come out through the rectum and the anus, as opposed to coming back out through the oral cavity, right? Or in some cases, the pathogen may get into the blood and then be eliminated, say, in the digestive system or in the urine, right? One of the properties that makes it possible for a pathogen to infect more hosts is something that's called latency. And that's the state where the microbes are inside the host and not causing disease, but could at any moment generate enough microbes in order to make you sick. Okay. An example is herpes simplex or zoster. Okay. And they have something called viral latency, where they actually have their genetic material inside host cells, either integrated into it or existing as a separate molecule. And it doesn't do anything until certain conditions are met and we start generating now new rounds of virus. Okay. So, for instance, if you had chicken pox as a child, the information from that chicken pox infection is still inside your body and could burst forth at any time, given the correct conditions, especially in an adult. As the immune system weakens, it sometimes pops out as a condition known as shingles. Okay. Bacterial or protozoal latency is seen in things like syphilis and typhoid and malaria. Okay. Remember that bacteria and protozoans have the ability to form these sort of sleeping states, right? The bacteria can form spores in some cases, right? And the protozoan can form something called cysts, okay? And they can just hang out until the right time to burst forth again, okay? Sequelae are long-term damage to tissues or organs that are caused by an infection. So, in other words, once you wage the war of the infection and maybe you eliminate the pathogen, what's the lasting damage? In meningitis, it can be deafness, strep throat, it can be heart valve disease. In Lyme disease, it can be arthritis and autoimmunity. In polio, it can be paralysis. Okay. And unfortunately, a lot of these changes, many of these changes are irreversible. Triangle means change. So once they happen, that damage is there forever. Okay? Inside your body, the pathogen is going to have an incubation period. And that's the time from when you first run into it to the time when the infectious agent generates symptoms. Hold on. Okay, sorry about that. So the time from when you first run into the pathogen to the time where the symptoms first pop up, okay? During this time, the agent is reproducing at the portal of entry, but it hasn't generated damage that causes the symptoms yet. The amount of time for the incubation period is going to be different depending on how resistant the host is, what the dose of the pathogen was, right? And how effective it is at producing the disease, as well as the distance between the target and the portal of entry. Okay. And during this time, you can spread disease. Okay. You can spread disease at this point. Okay. Which makes it a real dangerous time, right? The longer the incubation period, longer the incubation period, okay, comma, the more hosts get infected. Why is that? Because you're wandering around in population, not knowing you're sick, interacting with these people, and you've got stuff growing on you and in you, right? It ranges from several hours to several years, depending on the pathogen. Most infections range from 2 to 30 days. In the prodromal stage is when the earliest symptoms of an infection appear, okay? You get vague discomfort, head and muscle aches, fatigue, upset stomach, you get tired, right? So this is when you're first aware that you're sick. Okay. You're first aware. Then you've got the invasion period, right? The pathogen multiplies at high levels. You get a lot of toxicity and it gets established in the host. You'll see fever and specific signs and symptoms of the disease. And this is very variable as well. But here again, you are aware that you are sick. Okay. So you can see that the time between first contact and the prodromal stage, the longer that is, the more you can spread the condition. In the convalescent period, the patient is attacking the infection and symptoms get less and less. Patient's health and strength return because of the healing nature of our immune system. And a lot of patients stop taking antibiotics during this time, even though pathogens are still in our system. And this leads to antibiotic resistance. Why? Because from first contact, okay, through prodromal, all right, you can spread and feel well. And then in convalescent, okay, you may not know if you are infected. Okay. So these are dangerous times, right? Because you're out there feeling good, right? But you can spread it to other people. The transmissibility is going to depend again on the pathogen. In the incubation period, measles virus can be released, right? In the invasive period, Shigella can be released. And in all periods, hepatitis B can be released. Okay. So if we go through, right, the time when you're aware that something's wrong, it's going to be from here to here, right? You feel sick. Okay. Here, you feel better. Or maybe you feel fine. And then over here, you feel healthy. So the longer these periods are, incubation and convalescent, the more potential there is to spread the disease. Okay. And then, of course, in subclinical people, they'll spread it in all stages because they never feel sick. Okay. Even though they've got the pathogen and it's growing on them and in them. Okay. Where can pathogens hang out in the environment and keep getting us sick, right? So that's what we call a reservoir. Okay. It can be a human or an animal carrier. It can also be in soil, water, and plants. The transmitter is the object where you get the infection from. For instance, in syphilis, the reservoir and the transmitter are the same. They are the human host. Okay. And in Hep A, the reservoir is a human and the transmitter is food. Okay. So different way to get from host to host. Okay. A lot of infections that hit humans have reservoirs in other humans. Persons or animals with symptomatic infection are obvious sources and easy to avoid. But a carrier is someone who has the pathogen and spreads it to others and doesn't realize it, and who may not have any symptoms. Okay. Examples, right? You can be an asymptomatic carrier of gonorrhea or genital herpes without lesions, or human papillomavirus, right? You could be an incubating carrier of mono or a convalescent carrier of Hep A, right? or a chronic carrier of typhoid or tuberculosis. Okay. And in all of those cases, you're going to feel fine, right? Most of animal reservoir agents are things like fleas, mosquitoes, flies, and ticks, insects. Okay. Biological vector participates in the life cycle of the pathogen, so it provides a time during which it can complete its life cycle, while a mechanical vector is just like a car or a bus carrying the pathogen to the next location. Large animals can spread infection, such as raccoons or dogs or cats with rabies. Okay. Birds with pisticosis, lizards with salmonella. Okay. I got salmonella from a pet store turtle when I was young. Okay. And that was a mechanical vector. Because I would pick the animal up, I'd play with it, I'd put it back. And even if I wash my hands, sometimes there can still be bacteria there, and then it gets into your digestive system, and then you get the condition. Okay. A zoonosis is an infection that is normally found in animals but can get to humans, and they're going to be a dead end host and not spread the microbe. Some of these infections are going to involve many hosts, and some have very complex life cycles. So the spread of the disease is the result of the association between the human and the animal. And so you can see some examples here, right? We can take a few that we have in the area and point them out. Let's take rabies, right? Rabies, you come into contact with a rabid animal, okay? And you get it from a bite or a scratch. How can you tell a rabid animal? Well, if it's a wild animal, it won't run when you approach it. Okay, it'll come towards you, which is abnormal. Okay, if it's a domestic animal, it'll be probably agitated or angry. Okay, where normally it wouldn't be. Right. No cure for this, unfortunately, only a vaccination, which you have to get very quickly after the bite. Okay. Rocky Mountain spotted fever is big around here, especially in the spring, fall, and the summer. And that's ticks. Okay. So what does a tick do? A tick hangs out on plant life, and it can tell when you're approaching because it can tell the change in temperature. What he'll do is he'll drop off of a leaf or a branch and onto you, and you won't realize it. And then he'll secrete a numbing agent onto your skin, okay, and then burrow his head under your skin and into the blood supply. And when he takes his blood meal, he will give you the spirochete bacteria. That causes things like Lyme disease. Okay, where's Lyme disease on here? Specifically listed. Okay, or he can give you the bacteria that causes the spotted fever. Okay, and without that, these diseases would never spread. Okay, that's why you have to check yourself for ticks after you do yard work or after you are out in the woods in wooded areas, right? Other examples, ringworm. Okay, you can get this from dogs and cats, and ringworm is a fungus. the skin fungus. Okay. It makes these worm-like patterns as it grows on the skin and causes inflammation. Okay. And you treat this with antimycotics, which are antifungals. Antimycotics. Okay. So those are just a selected few, right? Non-living reservoirs would be things like the soil and the water, okay, or a surface, okay. Some are opportunistic and some are regular pathogens, right? Because humans are in contact with these sources, you can pick up the pathogen, and that tells us something about where the disease is coming from and how to prevent it, right? So that's what an epidemiologist does. Okay. Where is the disease coming from? Okay. A communicable disease is one where an infected host can transmit an infection to another healthy host, and it can cause the disease. Okay. Infectious and communicable mean the same thing. A contagious disease is one that is mostly communicable, especially through direct contact, such as flu and measles. Okay. Hansen's disease is weakly communicable. Okay. This is leprosy. And a non-communicable disease is one that doesn't arise through transmission of the infectious agent from host to host. A compromised person gets invaded by their own pathogens that are always around them or in them. Okay. The individual has contact in some cases with a microbe that's in a non-living reservoir. Infected persons don't become a source of disease to healthy people because their immune system can fight it off. Okay. So how can we transmit? Well, you can have direct or indirect contact with an animate or an inanimate object, right? An animate object is a living object. Okay. Horizontal transfer is spreading the disease through a population from one person to another, while vertical transfer is transmission from parent to offspring. Okay. Two major modes of transmission: direct contact and indirect transmission where there's a vehicle, right? Such as, say you have a cold, you sneeze and you sneeze into your hand, and then you put your hand on a doorknob and leave the room, right? Well, if somebody else touches that doorknob and then touches their face, then they can pick up your cold, okay? That's one of the reasons why we get more colds in the winter, is because we're inside more often and in contact with those vehicles more easily. Okay. So you can see some examples of the different types of transfer, right? The one that we mostly think about is direct contact, right? But actually, it's indirect transmission that's often more dangerous because we have to identify where the problem is coming from. Okay. And then we have what are called nosocomial infections, which are picked up in a hospital environment or a clinical environment. Okay. About between 0.1 to 20% of admitted patients pick these up, with an average being around 5%. There's between 2 and 4 million cases annually, with 90,000 deaths, 8 million in additional days of hospitalization, and an increased cost of between $5 to $10 billion to the healthcare system. So what causes this? Patients who are immunocompromised, all right? So say weak immune system. Okay. Collection point for pathogens, right? Which means lots of pathogens in a small area. Which means if you're there, you're more likely to pick it up. A lower defense permits normal biota to enter the body, okay? And so they can cause disease if they're growing where they're not supposed to be, such as an E. coli infection in your blood, okay? Normally the E. coli in your colon are good for you, but if those E. coli get into your blood, then you have septicemia. Infections can be acquired directly or indirectly from a fomite, which is an inanimate object, okay? From medical equipment, from other patients, from medical personnel, from visitors, and of course from air and water. The healthcare process can lead to these infections, such as treatment using reusable instruments like respirators and thermometers. That's why we try to make as many of those parts disposable as possible. Indwelling devices like catheters or prosthetic heart valves or grafts or drainage tubes, okay, allow bad stuff to get in. Increased proportions of the hospital population getting antimicrobial therapy, causing drug-resistant microbes that get selected for at a higher rate, okay? So one of the big players in this is something called MRSA. Okay, probably heard of MRSA infections. That stands for methicillin resistant Staph aureus. Okay, and you see it all over clinics and hospitals, and it basically causes a staph infection that you can't treat with the family of penicillin drugs, and sometimes with other types of antibiotics as well. Okay, very bad infection. The most common healthcare associated infections are UTIs, respiratory tract infections, and infections at surgical sites. Gram-negative intestinal biota are cultured in more than half of the patients with nosocomial infections. So again, these are microbes that are already in their digestive system. Okay. Gram-positive bacteria like staph or strep and yeast make up the rest. Okay. True pathogens such as Mycobacterium tuberculosis, Salmonella, and so on can also be transmitted. Okay. But the vast majority, notice, are these, let's get another color here. Are these urinary tract infections, UTIs. Okay. And so that's coming from MRSA. Okay. And regular staph and things like Pseudomonas. Okay. Control of these infections. Well, Medicaid and Medicare no longer reimburse for catheter-related UTIs, vascular catheter-associated infections, or surgical site infections. So the infection control officer is going to put into place practices and procedures that'll track the outbreaks, ID problems in technique, and train the healthcare workers to avoid these problems. Nurses and caregivers are usually exposed to needle sticks, infectious secretions, blood, and physical contact with patient. So they need to be aware of infection control. All right. And so that's our next topic here.
Touching hearts and strengthening lives since 1904. Bethesda Lutheran Homes and Services Incorporated provides supports, resources, and worldwide outreach to people with developmental disabilities.
Workers in many different occupations are at risk of exposure to infections. Examples of those at risk are people who work in housekeeping, maintenance, laundry, direct support, or anyone in the healthcare field. To minimize the risk of infection, it is important that everyone follows standard precautions. To understand what standard precautions really means, let's take a look at how the Centers for Disease Control and Prevention define standard precautions. Standard precautions represents a system of barrier precautions to be used by all personnel for contact with blood, body fluids, secretions, excretions, non-intact skin, and mucous membranes, regardless of the person's diagnosis. This system includes the concepts of body substance isolation, which reduces the risk of transmitting pathogens from moist body substances, and universal precautions, which reduces the risk of transmitting bloodborne pathogens. There is a rising concern about exposure to blood due to bloodborne pathogens. You're probably asking yourself, what is a pathogen? Pathogens are disease-causing microorganisms that are found everywhere in the environment, on humans, and animals. Pathogens found in the blood are called bloodborne pathogens. Some examples of bloodborne diseases are hepatitis B, hepatitis C, HIV, and malaria. Standard precautions is an approach to infection control that treats all human blood and certain body fluids as if they are infectious. Since these pathogens are too small to be seen by the naked eye, and you can't tell by looking at an individual whether they have a disease, you should protect yourself with simple and effective barriers. This is the rationale of standard precautions. Transmission-based precautions, in addition to standard precautions, are used when someone is suspected of being infected with a highly contagious pathogen. Transmission-based precautions are required where airborne, droplet, and contact transmission of infectious organisms may occur. Transmission-based precautions would be used if a person has chickenpox, measles, or an infection, to name a few. It is not always possible to know whether or not a person is infected with an infectious disease. Universal precautions is made up of a system of safeguards designed to help protect you and the people you support. Safeguards are things like personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccinations. All of these systems should be used together to form a protective barrier. We'll discuss these safeguards in more detail later. There are some people who may have an infectious disease and not show any symptoms or even be aware of their infection. For example, how many of you know the story of Mary Malone, also known as Typhoid Mary? Mary was the first person found to be a healthy carrier of typhoid fever in the United States. Typhoid fever is a life-threatening illness caused by the bacterium Salmonella typhi. You can get typhoid fever if you eat food or drink beverages that have been handled by a person who is shedding Salmonella typhi. Mary earned her living as a cook for private families. When several outbreaks of typhoid fever developed in homes where she worked, the public health officials questioned her. Under questioning, she stated she rarely washed her hands when cooking and felt it wasn't needed. She also insisted she never had typhoid fever and had always been healthy. However, the health officials linked the outbreaks back to her. After her death in 1938, the autopsy revealed that her gallbladder was still actively shedding typhoid bacteria. This example helps us to understand the importance of following standard precaution procedures at all times. Somehow, pathogens need to get inside your body to cause an infection. Let's take a few minutes to discuss how these pathogens can get inside your body. It can happen from a sharp object contaminated with the infectious material, such as a needle, broken glass, wires, or any other object that can break or cut your skin. From openings in your skin, this would include cuts, nicks, abrasions, skin infections, acne, and surgical incisions. Through your mucous membranes. Mucous membranes are the moist surfaces of your mouth, eyes, and nose. By touching contaminated environmental surfaces and transferring the contaminant to mucous membranes or open skin, surfaces can be heavily contaminated without visible signs of contamination. Diseases that are transmitted from other people or animals are called communicable diseases. Some examples of communicable diseases are measles, strep throat, and colds. Standard precautions should be used in the care of all people and used regardless of whether or not the person is known to have a communicable disease. In order for a disease to spread to others, certain conditions are needed. This is sometimes called the chain of infection. Let's take a few minutes to discuss these conditions. First of all, there must be an agent like a virus, fungus, or bacteria that can cause the illness. The agent also must have a place to live. This is called a host. Humans, animals, and insects are the most common hosts, although agents can also live in the environment. The agent must also have the right environment to survive. Often, moist, dark places help them to live and grow. The agent must be able to enter the host. For example, a person who has a break in their skin or someone who breathes in the agent. An agent must have a way to move from its host to infect others. This is called transmission. Some examples are through blood, body fluids, a sneeze, or a cough. There also must be a person who can catch the disease. If someone can't fight off the germ, they can become infected. This is called being susceptible. This completes the chain of infection. The cycle continues as the agent is passed on to another host. The easiest way to remember how diseases can spread is through the David method. Communicable diseases are mainly spread in the following ways. Direct. This is through person-to-person contact. Airborne. This simply means through the air. These infectious droplets remain suspended in the air because they are carried by microscopic droplet nuclei or dust particles. They can land on or be inhaled by the host. Vector. This occurs when an organism, such as a tick, carries disease-causing microorganisms from one host to another. Indirect. This occurs when microorganisms are transmitted by contaminated items such as food, water, medication, and equipment. In other words, person, object, person. Droplets. When someone coughs, sneezes, or talks, tiny droplets may be generated. These infectious droplets propel a short distance through the air and land in the host's eyes, nose, or mouth. Droplets do not remain suspended in the air. The spread of infectious diseases can be stopped by killing the agent, changing the environment so the agent can't survive, wearing protective gear, removing the agent's means of spreading, and making sure people are immune to the agent through immunization. This is commonly called breaking the chain of infection. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Hand washing is often taken for granted because it can be routine, but good hand washing stops the spread of contaminants from your hands to other parts of your body or other surfaces you may come in contact with. Let's take a few minutes to go over some general guidelines to follow when washing your hands. If you remove your gloves, wash your hands thoroughly. If skin comes into direct contact with blood or other infectious material, wash with soap and water as soon as possible. If you are using antiseptic towelettes or hand sanitizers, you should still wash your hands as soon as possible after touching something that is visibly contaminated. Remember, good hand washing doesn't take a lot of time. Follow these 6 steps for good hand washing. Remove any jewelry you are wearing if possible before washing your hands. Bacteria can thrive under rings and watches. When washing your hands, use warm, running water. Wet your hands and apply soap. Use plenty of soap to create a good lather. Vigorously rub together all surfaces of your hands for 15 to 20 seconds. Friction helps remove dirt and microorganisms. Pay close attention to cuticles and under your fingernails. Keep splashes to a minimum and try not to touch the sink itself. Rinse your hands, keeping your hands lower than your elbows. Dry your wrists and hands completely with a clean paper towel. It is best to pat your hands dry rather than rubbing them. This helps prevent your hands from chapping and cracking. Turn off the faucet. Remember, don't touch the faucet with your clean hands. Use a clean paper towel. Remember these 6 steps the next time you wash your hands or assist the people you support to wash their hands. You're probably now asking yourself, how often should I wash my hands? The following are general guidelines for washing your hands. When you arrive at work, before and after assisting someone with personal care, before and after wearing gloves, before and after you eat, before and after cooking, before leaving work, after blowing your nose, touching your face or hair, after using the bathroom, after any cleanup you do, after smoking, or whenever your hands are soiled. Standard precautions requires the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids, such as mucus, drainage, or emesis. Personal protective equipment helps protect you from contact with blood or other potentially infectious material. Personal protective equipment includes gloves, masks, gowns, aprons, face shields, protective eyewear, mouthpieces, and resuscitation bags or other ventilation devices. Personal protective equipment must be appropriate for the task. You should wear as much as you anticipate you will need to keep blood and other infectious materials off your skin, mucous membranes, or clothing. Anytime the personal protective equipment is torn or contaminated, remove the equipment and replace as soon as possible. Gloves should always be used whenever you anticipate contact with blood, potentially infectious material, mucous membranes, or non-intact skin. When working directly with people, single-use disposable gloves should be worn. Single-use disposable gloves are just that. They should be used only once and then disposed of immediately. The wearing of gloves does not substitute for good hand washing. It's not acceptable to wear the same gloves or wash gloves between each person. Another type of glove you may use is a utility glove. Utility gloves are generally used during housekeeping. Utility gloves should be cleaned after every use with soap and water and then dipped in bleach solution. The gloves should be worn, not handled, during this cleaning and sanitizing procedure. Utility gloves are not used when working with people They are only appropriate for cleaning. Because gloves can be torn or damaged, always bandage any cuts or open areas on your hands before applying gloves. Replace the gloves as soon as possible if they are contaminated, torn, punctured, or damaged in any way. Follow these general guidelines to determine when to wear gloves. Anytime you are handling blood or body fluids. Anytime you have open wounds on your hands. Anytime there is potential to come into contact with objects contaminated with blood or body fluids. If you are touching mucous membranes, when touching non-intact skin of any person, anytime you are cleaning contaminated or potentially contaminated surfaces, equipment, or objects. Good housekeeping helps protect everyone and is everyone's responsibility. Carefully follow your agency's policies and procedures regarding handling infectious materials. The following are some general rules when dealing with infectious materials. Always wear gloves when handling infectious materials. Handle used equipment soiled with blood or other potentially infectious materials with care. Do not let the equipment touch your skin, mucous membranes, clothing, or other items in the environment. Do not eat, drink, smoke, apply cosmetics or lip balms, or handle contact lenses where there is reasonable chance you have been exposed to blood or body fluids. Do not keep food and drink in places where blood or other potentially infectious materials are present. Clean reusable equipment appropriately before using it for another person. Single-use items should be discarded. Be careful to prevent injuries from needles and other sharp instruments when using, cleaning, or disposing of them. Never bend, recap, or break needles after use. Dispose of contaminated sharps immediately after use in appropriately labeled or color-coded, leak-proof, and puncture-resistant containers. Report any container that is full. Remember, safety razors and razor blades are also considered sharp material. Follow your agency's guidelines when disposing of sharp materials. To prevent contamination, handle soiled linen as little as possible and with minimum agitation. Gloves should be worn. Without sorting or rinsing, place soiled linens in a leak-proof bag in the area where it was used. Follow your agency's guidelines for handling contaminated linens. Transport specimens in closed containers. Wear gloves and handle the containers carefully. Specimens taken to the doctor should be placed in a closable plastic bag. Carry waste containers away from your body. Clean all blood and body fluid spills promptly according to your agency's policy. Everyone should know what to do if an accidental exposure of blood or body fluids occur, such as a needle stick injury or a splash into the eyes or mouth. This is called a significant exposure. If you are involved with a significant exposure, don't panic. Immediately wash the affected skin with soap and water. Flush affected mucous membranes, such as eyes, with water. Report the incident to your supervisor immediately. There are a number of steps that must be taken in the first 24 hours to decrease your risk of becoming ill, so quick reporting is essential. Follow your agency's guidelines when dealing with any accidental exposure to blood or body fluids. If there is a blood spill, your agency may have a blood spill kit for your use. Follow your agency's policy for using your specific kit. Infection control practices are easy to ignore because they can be so basic, but you must take the time and make the effort to consistently follow infection control procedures. Let's take a few minutes to summarize some of the main points when following standard precaution guidelines. Pathogens are disease-causing microorganisms that can be found everywhere in the environment, humans and animals. Standard precautions is an approach to infection control that treats blood and body fluids as if they are infectious. Standard precautions includes the concepts of universal precautions and body substance isolation. It is not always possible to know whether a person is infected with a disease. The person may not show any signs or symptoms or even be aware of their infection. Standard precautions are made up of a system of safeguards designed to help protect you and the people you support. These safeguards include personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccination. All must be used together to form an effective barrier. Pathogens need to get inside of your body to cause an infection. Some ways they may enter your body are by punctures from sharp objects or openings in your skin. Pathogens can also enter through mucous membranes of your mouth, eyes, nose, or by touching contaminated environmental surfaces. In order for a disease to spread to others, certain conditions are needed. These conditions include an agent, like a virus or bacteria that can cause illness, a host, a portal of entrance, a means of transmission, and a susceptible person. Transmission of pathogens can occur by direct, airborne, vector, indirect, or droplet means. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Standard precautions require the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids. Following standard precaution guidelines is crucial for protecting you and the people you support from potential infection.
The next topic we're going to address is Koch's postulates. And these are something that came out after the microscope was invented in the late 1700s that allowed us to try to assign an identity to a pathogen. Okay, and it wasn't possible until the advent of the microscope to do this because we couldn't see the pathogens. So a causative agent is the cause of the infection and the disease, right? And Koch's postulates are just a series of steps that you go through that allow you to assign the cause of an infectious disease to a particular organism, right? And it's still important today. It's reliable for a lot of diseases, but there are some situations where you can't use it, okay? And these include some pathogens that can't be isolated or grown in the lab, and some infections that you can't cause in humans, okay? That can't be produced, for instance, in animals, right? Viruses, for instance, have a limited host range, and human viruses only hit humans, and swine viruses only hit swine, and so on, okay? And then in some cases, if we have a biofilm that's causing the disease, we can't really determine the exact cause, right? Because a biofilm is a community of different microbes working together to produce this condition, and you can't really assign the disease to any one participant. You just sort of have to say it's the fault of this population of different microbes, okay? So the first thing that Koch's postulates requires is that you find evidence of a microbe in every case of a disease that you've managed to isolate. And you isolate that microbe from the infected subject and grow it up in a lab, okay? And then you inject that microbe into a healthy host that is susceptible, right? And they get the same disease symptoms. And then you're able to then sample that organism and re-isolate that microbe and identify it in every case where the test animal got sick. Okay. And so the steps where you use microscopic observation, right, were things that we couldn't do until the invention of the microscope, right? So these two steps here came after the 1800s. Okay. We'll talk about epidemiology and identify different kinds of epidemics. We'll talk about the curves that go with each one. So epidemiology is just a study of the frequency and the distribution of disease and other factors in particular populations. And it uses a variety of different scientific disciplines to try and establish how it is a disease can spread in a particular area. Okay. It considers all forms of disease. All right. Florence Nightingale was one of the people that laid the foundation for modern epidemiology. Before the discovery of the germ theory of disease, she understood that filth was associated with disease. And so she instituted methods in the military hospitals that included separate linens and towels for each patient, and the cleaning of floors and the unclogging of sewer pipes. And she kept notes and demonstrated that more men died of disease than of traumatic injury. Okay. And we now know today that was due to infection in the facility. So what do we do in epidemiology? We track behaviors like exercise and smoking. We use clues on what might be causing the infection. We use pathology, and we use modes of transmission to provide clues. We track the number and the distribution of cases in a community, and the outcomes of these studies allow us to help public health departments develop prevention and treatment programs and to predict how it is that the disease will spread and know how they can stop it. Okay. So a reportable disease has to always be reported to the authorities, and other diseases are voluntary, right? There's a lot of agencies at different levels that track infectious disease, and there's a form or table in your book 11.10 that gives you that list. Statistics of concern to the epidemiology of a disease include the mortality rate, which measures the total number of deaths in a population due to a disease, and the overall death rate from infectious diseases has dropped, although the number of persons afflicted with the infectious disease can go up. Okay. And that's through history, right? So this is called the morbidity rate, right? Morbidity rate are those that are afflicted and added to those that are dead. Okay. So death plus infections with symptoms. Okay. A point source epidemic are infections that came from a single source and all the victims were exposed to it. A common source epidemic comes from a common exposure to a single source of infection over time. And a propagated epidemic comes from an infectious agent communicable from person to person, which is sustained in a population. Okay. So these are usually limited scope and time. Okay. These have broader scope. Okay. And then these have broader scope. And more time. Okay. The index case is the first reported case. And that's the first one that the investigation picks up. Okay. It may not be the first case of the disease, but it's the first case that the officials are aware of. An endemic is just an infectious disease that has a steady frequency, right, over a long time in a particular area. Okay. Sporadic disease, these are occasional cases at irregular intervals at random locations. Okay. An epidemic is when stats indicate that the prevalence of an endemic or a sporadic disease is increasing beyond what we expect in that population. Time period's undefined, but it can be hours to years, and the exact percent of increase needed before an outbreak qualifies as an epidemic is not defined. And a pandemic, which is what we're in now, is the spread of an epidemic across continents, right? And pandemics are possible in the modern era because of transportation, okay? They were less likely before the invention of planes, trains, and automobiles. So you can see some examples here of an endemic, right, occurrence, an epidemic occurrence, see different areas, right? Sporadic, okay, and then pandemic, which is like what we have for COVID, okay, across many different continents, right? So the job of an epidemiologist is to try and figure out where the disease is coming from, what's causing it, right? And then suggest what measures to take to keep from spreading, right? For instance, if it's coming from contaminated water, you either purify or change the water source, okay? If it's airborne, then you're going to have to use techniques such as quarantine or filter masks, okay, in order to cut down on the spread and on the prevalence. Those are just a couple of examples, okay? All right. That's all I've got for today in lecture. We're going to continue with chapter 15 in lab. And in between now and lab, I'm going to get you your access codes. And I'm going to load your grades in. Okay. So you'll all know where you stand. All right. And in lab today, we are going to look at, let's pull it up here. Let's do a new share. We want that. Okay. And we're going to do a lab on water pollution. So what I will do is I'm going to generate an activity for you that I will send you later today. Okay. And then you'll complete that and email it back to me before we meet next Wednesday. All right. Keep in mind that I'm going to adjust the Connect site so that I only take your higher score on all of your attempts with your Connect exams and your Learn Smarts. All right. And there's no late penalty or anything that is associated with Connect because they didn't have access codes for us. Okay. So that will not be factored into the grade. Okay. So, Yeah, go ahead.
Are we gonna be able to see the video? That was-
You mean of this lecture?
Yes.
Yeah, yeah. It compiles after we stop, right? And then I load it up to YouTube.
Okay,
Yeah,
thank you.
yeah. Okay. So any more questions before we bump out and we meet again?
When you do the tutoring on Fridays, can you record those too?
Yeah, yeah, those are recorded. I can load those up. The
'Cause I
thing
never
about
saw
those is that
Access code for that.
the thing about the tutoring on Friday is that depending on who shows up, it's going to be different topics. Okay. If it's a tutoring session that is going to involve this class, then I will give you the link for the recording that relates to this class. Okay. The tutoring on Friday is like first come, first serve, and it's for any course I'm teaching or any course that the student needs help with. So they identify their school and their topic and their course, and I help them out with that. And then I record it and I give it back to them if they want to use it to study from. So if somebody from the class shows up, needs help, then I'll record that and I'll make that available to everybody.
Do you send out a Zoom access code for it?
Yeah. For the Fridays to come. Yeah. I'll give that to all you guys.
Is there a website to do your lab?
No, those are doing well for this activity. I don't know yet. I'm going to have to look around. Okay. For the previous two labs, there was a website and I sent that to you guys. So you have access to that.
I never got that.
Me
I'll
either.
resend it.
But you resend to Melissa Davenport too?
Yeah. Yeah. I'll send you all that stuff. So does anybody have any questions other than what we've already asked?
And you said our access code gonna be in our
I'm
email?
going to send those out right after we're done.
Okay,
Yeah.
thank you.
Okay. All right, y'all. I'll see you in lab.
Yeah.
Yeah.